Our second presenter today is Radhika Malik, who's an assistant professor at the Icahn School of Medicine at Mount Sinai, also in the Agarwal group. And today she'll be talking about cryo-EM structures of DNA polymerase delta and translesion DNA synthesis. So
okay, thank you.
Your slides are up and I can see full screen.
Okay, great.
Thank you again for being here and presenting.
Thank you for having us. So basically, Rinku gave a great introduction about high fidelity polymerases. So what I'm going to talk about is when these high fidelity polymerases encounter DNA damage. These are basically then replaced by these Y family, highly specialized Y family polymerases, which are mostly single subunit polymerases, and then DNA polymerase Zeta, which is a multi-subunit polymerase, similar to DNA polymerase delta. So we're going to talk about polymerase Zeta today. So our cells encounter DNA damage from various external sources, such as chemical carcinogens, ionizing radiation, and ultraviolet light every day. And also from internal sources, such as hydrolysis and oxidation. So one central mechanism which our cells is able to use is translesion DNA synthesis to battle these damages. So once a high fidelity replicative polymerase gets stalled at the DNA lesion, there's a two-step process which follows, wherein a Y family polymerase such as DNA polymerase iota would come in and put in a base opposite the lesion, which is followed by DNA polymerase Zeta coming in and extending beyond the mismatched primer termini. So once DNA polymerase Zeta has done its job, it basically leaves, and then the high fidelity polymerase takes over and carries out the normal DNA replication. So what is DNA polymerase Zeta? It's basically a crucial enzyme that allows our cells to battle DNA damage. It's a member of the B family of polymerases capable of translesion DNA synthesis, wherein it basically acts as an extender past DNA mismatches and lesions. In terms of the composition of Pol-Z, it's made up of 4 subunits, Rev3, Pol31, Pol32, and Rev7. Rev3 has a catalytic module and 2 cysteine-rich modules at the C terminus called cis-AD and cis-BD. Pol-Z has Pol31 and Pol32, which it shares with Pol-delta. Pol32 has a PIP motif at the C terminus, which interacts with pCNA at the site of DNA damage. So the third accessory subunit that Pol-Z has is actually unique in all B family polymerases. It's a member of the HORMA family of proteins, which basically have other functions in the cell, such as spindle assembly checkpoint proteins during chromosome segregation. Rev7 basically interacts with the RIR domain of Rev3. Rev3 has a polymerase active site and an inactive exonuclease site. Rev3 has one binding site which interacts with Rev7. So for a long time, it was thought that it was a 4 subunit complex. But recently, a second site in Rev3 was identified, which led to the incorporation of Rev7. In fact, in our structure, we have captured for the first time a dimeric Rev7 structure in the holoenzyme. So first of all, we started off with the purification of Pol-Z, where we were able to get a stoichiometric complex of the 4 subunits. Then we used this sample for cryo-EM studies of the ternary complex with the DNA and the incoming nucleotide. The first set of data that we collected at 0° stage tilt led to an anisotropic map. And so we were looking at different sample preparation methods at that time and found that the one that worked best for us was Chameleon. Chameleon is basically a robotic plunge freeze system which uses self-plotting nanowire grids. And the cool thing about it is that you can vary the spot to plunge times, which enable you to overcome the preferred orientation problem. So we used the data collected on these grids and then picked particles with a new neural network-based program called Topaz, which helped us in finding side views such as this and other orientations which we were not able to see before. We did further processing in Cryospark, which gave us a good ab initio model, and then a final reconstruction to around 3.2 angstrom with a good sphericity. We also did Cryo-EM studies of the Apo-state of Pol, where we were again seeing a similar problem of getting anisotrope maps. So for this one, we basically tried tilting here, and we tilted the stage to 40 degrees to get different orientations of the molecule. The data collection was very tedious and the processing was also quite challenging. However, ultimately we were able to get decent 2D class averages where we could see these side views and also other orientations, which helped us in building the ab initio model to about 4.1 angstroms with a sphericity of 0.92. So the structure of Pol Z looks like a ring. The DNA is in red, which basically contacts only the Rev3 in cyan. The CTD of Rev3 has a 4 iron 4 sulfur cluster shown here in brown. Apart from the core of Rev3, there is these motifs which extend out and weave around the two Rev7 molecules, and this is called the Rev3 RIR domain. So this is basically a map which has the same color code as the previous slide. And one of the things that we did not see in our structure was the C terminus of Pol 32, which we think would become ordered once it interacts with pCNA. So as you can see in the structure, it's a pretty flat molecule with a width of 60 angstroms. I have colored the subdomains of Rev3, and here you can see that the exonuclease and the cis-BD interacts with the Pol 31 subunit. And then you see these conserved RBMs, part of the RIR domain, interacting with Rev7A and B respectively. Since the structure of Rev3 was a novel one, we wanted to look at in further detail. So once we look at it, we see that it's actually quite similar to a typical B family polymerase, but there are some key differences. The NTD, which bridges between the exonuclease and the fingers domain, is much more extended in our structure. The fingers domain actually has an extra helix in comparison to B family polymerases, which does not interact with the nascent base pair, but in fact interacts with the NTD. The palm is much more extended, which we'll talk about later. The thumb interacts with the duplex portion of the DNA. The cis-BD is ordered and has this 4 iron 4 sulfur cluster, but in our structure of cis-AD is disordered. The RIR domain and a part of it basically interacts with the thumb domain and the palm domain. So when we zoom into the active site, we see a very similar architecture to the B family polymerases, where the active site residues come in from the fingers domain and the palm domain. The fingers are in a closed conformation, which impinge on the incoming T0 P0 base. Also, we see these active site metals and then the residues D975 and 114 coming in from the palm domain. So what we wanted to do was to look at this active site and compare it with the active site of DNA polymerase delta. So once we compare these two active sites, we find they are strikingly similar, which basically tends us to believe that the features that impose fidelity during the nucleotide incorporation step is quite similar with the high fidelity polymerase. So this was what we see around the T0, P0 side, but upstream at the T1 side, the story is much different. So the T1 side is the place where the lesion would come in once a Y family polymerase has put in a base opposite it. So in Rev3, what we see is a large pocket around this T1 base. So this is illustrated in the next slide, where the DNA is in red, the Rev3 is in blue, and Pol3 is in gray. What we want to focus on is this linker which connects the entity to the palm, which we call the NTD palm linker. In the case of Pol3, you see that this linker is very close to the T1 base. However, in case of Rev3, this loop has been pulled over by the helix as well as a palm loop, which is a unique structural element in the palm domain of Rev3. If we zoom into this area, we see that the tyrosine stacks against the sugar at the T1 site in the case of Pol3, whereas in case of Rev3, you see that equivalent residue glutamate being pulled out, creating ample space at the T1 site for a lesion to come and bind in. So this is basically the structural basis of the extension reaction, which Paul Z is very famous and does this very well. So what I'm going to do now is switch gears and talk about the Rev7 dimer. As I told you before, that Rev7 is part of the family of HORMA proteins, such as this MAT2. Usually these proteins exist as dimers. They can exist as open closed dimers or closed dimers. Closed dimer means there's a substrate which is bound to the C terminus of one of the molecules, and this is colored in cyan and it's called the safety belt region. These dimers are arranged in a head-to-head arrangement where these helices, these C helices, form a symmetric interface. Whereas the dimer that we see in our structure, in our holoenzyme, has these C helices separated further apart. And actually, the interface is asymmetric, where the helix C interacts with the safety belt region of the other monomer. So in fact, the Rev7 dimer is the organizing center of the Pol Z holoenzyme, wherein the Rev7b would interact with the phosphoesterase domain of Pol31, the N-terminus of Pol32. The Rev7a exclusively interacts with the RIR intermediate region. And then we have these motifs, RBM1 and RBM2 interacting with Rev7a and Rev7b respectively, forming a closed conformation. We have these prolines which are conserved between the RBMs and between yeast and the human form. And so we think that this type of an interaction would be conserved in all other Pol Z eukaryotic species. Also, we think that our Rev7 is an adapter protein which would basically attract other factors in the TLS machinery. So we wanted to compare the Pol Z with the Pol delta holoenzyme because both of them are from the same family, the V family of polymerases, and also they share Pol 31 and 32 as their accessory subunits. As Rinku mentioned earlier, that Pol 31 and 32 are quite flexible in the Pol delta holoenzyme. That's not the case in Pol Z. Is actually relatively rigid, probably because of its interaction with Rev7s. And this can be demonstrated by the local resolution map of the complex, which is has almost the same resolution across all subunits. Also, this rigidity can be attributed to an inactive exonuclease site, So unlike Pol delta, Pol zeta does not need to do proofreading. So we also had this Apo structure, and we wanted to compare it with our ternary complex structure. And the main difference we found was these fingers domain, which in case of the Apo form have moved almost 15 degrees outwards in comparison to the ternary complex. And so we think that the opening and closing of fingers augments Pol-Z's fidelity for the nucleotide incorporation step. So in conclusion, the structures of Pol-Z unveil a pentameric ring-like architecture. We propose structural elements such as the NTD palm linker, which will allow this unique polymerase to perform translesion synthesis. In the Pol-Z holoenzyme, we see two molecules of Rev7 arranged in a novel head-to-tail fashion, which has never been seen in the Horma family of proteins. In comparison to Pol delta, Pol zeta is a relatively rigid enzyme. And I didn't have time to go through it too much, but Pol-Z has emerged as a major drug target for chemo-resistant tumors. But the effort to target this enzyme have been severely hindered due to the lack of structural information on this enzyme. And so now we have these critical protein-protein interfaces, which can now be targeted for drug development and treatment of chemo-resistant tumors. So finally, I'd like to thank my advisor, Dr. Anil Agarwal. He has been tremendously supportive throughout this work. This work took a long time and a lot of back and forth. And so he has always been very supportive and encouraged us on the project. I'd like to thank Iban, Misha from the New York Structural Biology Center, and Jakub for help with the EM work, Rinku for help with model building, and then our collaborators in Texas for expressing Pol-Z, which is not very difficult to do. I mean, it used to be difficult to do, it's okay now, and the yields are okay for cryo-EM studies, not for crystallography really. And also our great advisors at the SEMC, Bridget, Clint, Ed, and Yongzi, from whom we used to get great advice regularly. And thank you for your attention, and thank you for giving us the opportunity to talk in SBGrid. Thank you.
Thank you, Radhika. That was also another very interesting talk. And again, for the folks in the audience, if you have questions, please feel free to send them to me in chat. We have slightly fewer so far. I guess one question to start with is, you compared the rigidity of the Zeta polymerase to the Delta polymerase a couple of times. Do you think that is that a reflection of the enzyme rate? Is that something just to do with the different types of DNA synthesis you're doing, or is it just a different observation that is not clear yet?
That's a good question. Certainly a different observation. And I think we need to further investigate that by doing, I don't know, some mutations and stuff. So yeah, it's just an observation for now.
That's where things start, but it's always curious. For your hollow complexes, it looked like the DNA that you have modeled in was relatively short as far as how far it extended out. Is that just because that was the length of the DNA that you put in the complex for assembling, or is there more DNA there that's just disordered and not visible?
Yeah, there is some part of the DNA. I think there are like 4 nucleotides which are disordered, and we did not build that into the model. Okay,
thank you. And a follow-up question for rigidity: is the rigidity a central feature of the TNase polymerase and polymerases in general?
Well, this was, so Pol-Z is very unique in the fact that it's the only multi-subunit TLS polymerase. first of its kind. All other TLS polymerases are single subunit polymerases, which only have a Pol active site and an exonuclease inactive site. So basically, there's no other real comparison. So we always compare it with Pol delta, which is the other high fidelity B polymerase. So yeah, it's quite unique in that way.
Thank you. We have a couple questions related to the grid preparation and the anisotropy. So I guess one of them is, do you have, if you had anisotropic problems with the preferred orientation, is that just, do you think that that's something that you could rationalize based on the overall shape of the molecule?
Yeah, so basically it's quite, if I can go back to my slide again. How do I do this? So, yeah, so if you look at the molecule, it's quite flat. And this orientation is only 60 angstroms. So we feel that that it could be lying on the grid. So it basically was lying on the grid in this orientation versus, and we could not find side views at all initially. And so we think that could be a reason, and that could be an explanation for it. I mean, you can explain air-water interface interactions as well, which usually is a problem for sample preparation during when you do it normally, normal plunge freezing. But yeah, we can say that the flat shape of the molecule could have contributed to it.
Thank you. Like I say, follow up, another chameleon-related question. Can you say like which protein concentration you used for chameleon grid freezing, and is that in the normal range for what you'd use without the chameleon?
So we did have to use about 10 times higher concentration that we use normally for our grid preparation. So I was doing around 0.2 to 0.3 mg for my normal grids, but for chameleon we had to go to almost 1 mg per mil to get a decent data set with the chameleon grids.
Okay. We have a question where I'm going to reveal my ignorance of experimental cryo-EM. Do you measure the ice thickness for your grid?
Yeah, we do measure the ice thickness while we collect our data. We use Leginon for data collection, and actually ice thickness calculations are incorporated during data collection for every image that we collect.
Thank you. That runs through all the questions I've seen so far. So.
Okay, great. Thank you.
Thank you for presenting, and for everybody joining in. Thanks for being here today. And we will see you next week, or at least we will be here next week. If you would like to join in, please feel free. So thanks again.
Thank you, everyone.
Thank you, everyone.
Bye.
Bye.